To start this second video, I just wanted to go over a quick review of our previous video. So in the previous video, we learned that cell division is the process of cells dividing, and it results in two identical cells. And ultimately, this is how we go from being an unborn fetus to a full-grown adult. It is simply through repetitive cycles of cell division. Next, we went over the hierarchy of life. All right, that's where an organism is made of organ systems like the digestive system, the respiratory system, the pulmonary system, and then each one of these systems is made up of organs, and then we just continue down this hierarchy all the way to cells. After that, we talked about cell differentiation and how cells each have a special function and a special job within the body, but there are certain types of cells called stem cells that aren't differentiated yet, and so they have the ability to become different cells, and there's a lot of promise in science in the future with stem cells and stem cell research. Then we started actually talking about a part of cell division, and that has to do with interphase. So interphase is the largest part of the cycle that we're going to be talking about. Now G1 phase, this is where growth, development, and protein creation takes place. The S phase of interphase is where the DNA of the chromosomes end up replicating and splitting. And the G2 phase is where we again have more growth, development, and protein creation. And then we ended off with some vocabulary, talking about chromatid. That's half of a chromosome, and sister chromatids are identical chromatids side by side in a chromosome. The centromere is the center where the sister chromatids meet up, and the telomere is where the chromosomes, the very ends of each part of the chromosome. Now, we are stepping foot into the kind of bread and butter of this unit with something called mitosis. And you're going to hear me say that word a lot. So mitosis ultimately just means cell division. And when we talk about that, it's always going to be one cell dividing and becoming two identical daughter cells. And again, remember, a parent cell is the original cell. Daughter cells are the two cells that are a result of mitosis. Now, mitosis can be broken up into a number of stages. So before I put any of the labelings on, I just want you to look at this. I'm going to kind of look at the progression from left to right. And we're going to do a lot of work with this. But this is the entire process of mitosis. So starting with the left, we have prophase. And then we have a step in between, because ultimately going from prophase to metaphase, there are a couple steps that have to happen. So prophase, all the way to the third one to metaphase, and then anaphase, telophase, and cytokinesis. Now I'm going to challenge you to come up with some sort of mnemonic device that you're able to remember this, because it's going to be crucial that you remember the order of mitosis. So prophase, metaphase, anaphase, telophase, cytokinesis. Now we are going to move on and talk about each individual stage. So, first phase, prophase. As you can see from the picture on the right, the chromosomes still look like chromosomes. They are, as opposed to interphase, they are in the stereotypical X pattern. They have become condensed, And when you see this X, we know that those are sister chromatids. Again, that vocabulary word that came up. Now, the nuclear membrane around the nucleus disappears, which means that the DNA is able to kind of move freely because, recall, DNA can't normally leave the nucleus. And then spindle fibers form out of our chromosome. So centrioles. The spindle fibers form out of the centrioles. So you can see in the top right of that cell, those are two centrioles. And recall, the sole purpose of that organelle is to help out with cell division, and we have come to that point right now. Our next step is metaphase. So as you can tell, the spindle fibers from the centrioles connect to the centromere of each sister chromatid. Now that's a lot of vocabulary words, so I'm going to say that one more time. The spindle fibers, which you can see kind of in that purple color coming from the centrioles, which are the kind of teal cylinders, are connecting to the center of our sister chromatids, which is called the centromere. Also, you might have noticed that all of our chromosomes are lined up in the center of the cell. And you'll see moving forward that is crucial that alignment occurs. Next, we have anaphase. So, we went from having an X, and now our sister chromatids are becoming separated. And so they now are considered chromatids. So if you were to draw a line all the way across, technically they're still identical. They've just been pulled to opposite sides of the cell. Next, we have telophase, and you can start to see that before the cell even divides, you're seeing two kind of cells starting to form within one. So the dotted line around our chromatids, that's our nuclear envelope. It reforms. So now we kind of have two nuclei, two kind of nuclear envelopes within one cell. The chromosomes start to look like chromatin again, so being thread-like instead of rod-like, so they're kind of condensing and going back to the original shape. And the spindle fibers break down and dissolve because they're no longer, their job is done. Their job was to line up the sister chromatid in the middle and pull them apart, and that is all complete. So the centrioles kind of go back to being dormant. And this is like our beginning stage of cytokinesis, and that's going to be our last step. So now we have cytokinesis. And if we were to break down this word, again, cyto means our cell, right? Kinesis means to move or to split. So this word means a division of the cytoplasm into two individual cells. In plant cells, it's a little different, right? We have the cell plate and cell wall. So the cell plate forms in between the two nuclei. Recall to telophase, where we have the two different nuclei on either side. So the cell plate forms and eventually turns into a membrane, creating our two different cells. In the animal cell, it's a little different. It forms something called a cleavage. Ha, 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 yes, ha, funny. But ultimately, cleavage is just where the cell pinch. It furrows, it pinches into two equal parts, and eventually that pinch closes off, and two cells are the result, two identical cells. So how often do cells divide? This is a great question, and the best answer I can give you is it depends. All right, every cell has a different time frame. So, for example, the internal lining of intestines, cells divide every 5 days. Okay, so that's fairly often. Skin cells, about every 2 weeks. So we're increasing the amount of time that it takes. Red blood cells, it's about every 4 months. So again, we're really starting to get up there. And liver cells, about 1 year. All right, so as you can tell, liver cells, if we damaged our liver cells, it takes a very long time kind of to replenish the stores. So the big question here is why? Why do we have to go through cell division? This seems like a really complicated process. Why do we have to do it? And if you take everything else away, it's all about growth and repair. Right, we've all been through a phase when we were younger where we grew a lot. The only way that's possible is through mitosis. We simply need more cells in our body. If we didn't, we would never be able to get bigger. We would never be able to grow. Our bodies would just stay the same size. Additionally, sometimes cells don't function quite right. And so we need to be able to get rid of the cells that aren't working properly and replace them with, properly functioning cells. One more thing, too, is that the individual cells, they grow as well, and they all reach a point where they simply can't grow anymore. It just doesn't allow for it. And so if the cell wants to continue to move things in and out at a reasonable rate, they need to be able to divide and keep their size and appropriate kind of volume and surface area. So this is a quick summary of mitosis. Again, this is very simple, but it's going to be crucial that you know the order, the the specific steps for each one, and why they're important.